that's what you wanted. So let's have a recap on secondary structure prediction. Again, let me repeat. As of yesterday morning, we were still looking for IDP students for this carry your gene concept. I do know that Lothar, who is on the last slide here, had been contacted and talked to a couple of people yesterday. I'm not sure whether it's true, but we're still looking for IDP anyway, maybe not for this one. Recap on secondary structure. So again, we take this protein sequence, stretch it out, throw it in solvent, and adopts a unique three-dimensional structure. In principle, therefore, the problem is entirely defined by the environment and the sequence. So in principle, we should be able to predict three-dimensional structure from sequence alone, if we understood the physics right. And that is the crux here. We don't quite understand the physics well enough. There are many, many problems. What we can certainly do as a simple method is to compare sequences. The simplest way to compare is to simply line up the letters in a way that they optimally superpose, fit. That's called dynamic programming. It's a brute force algorithm that has a problem in time. There are there are fast methods such as BLAST that help speed this up. Ultimately, all these types of methods compare two sequences and decide whether or not they are similar based on all that you see in the comparison of these two. That's called sequence-sequence comparison, pairwise comparison, and that is what you can apply when two proteins are relatively similar to each other. Top daylight zone. When there's a twilight zone where you lose the signal of similarity, you need to come up with new methods that now no longer only look at sequence-sequence, but at sequence profile. Profile means I build an entire family. Build means I take a family of protein, I just do what I did before, pairwise, and I find a lot of them. And finding a lot of them, I feel that they are all related. They're part of a family. Family just means they are similar to each other. That way I have a profile of similarity. Profile, essentially I know exactly in that family which ones are the most important positions. And that profile I can use to align a sequence into this. That has more information because now I no longer use only a pair of sequences, but one sequence against a bunch of others. And there's a lot of detail in that profile. Sequence profile methods allow us to go into this twilight zone, to go deep into the end of the twilight zone, into the beginning of the midnight zone, where really have no signal anymore, is where we need the profile methods for. Same idea, except now I have profiles on both sides. The good news is comparative modeling. That is the technology that I use when I have an experimental structure, a sequence of unknown structure. I see it has a similar sequence to one of the experimental structures. I look up the 3D structure. That is comparative modeling. Reliably predicts the structure for 25 million proteins, but the bad news is for most proteins it still doesn't work. You have to simplify, therefore, the problem. 3D works for around about half the glass, and then we have to go to 2D and 1D. I begin with 1D, strings, sequence here, secondary structure, solvent accessibility, and that is what we talked about. The first generation methods that simply look at single residue frequency, second generation that looks at the windows, what's the probability given the proline in the center of a window of, in this particular case, 7, or even more depending on how you count this window here. That one breaks the helix. There are problems with these methods. One is they're clearly less than 65% accurate. Beta strands are predicted at almost random levels, and segments are predicted too short. I showed you a machine learning device, neural network. The way it does it, in this particular case, 13 consecutive residues. Proline happens to be in the center. Each one of these blobs here is 21 consecutive units. 21, I need 20 for the 20 amino acids. 1 as a spacer, the 21st unit as a spacer, so that the central residue can be at the begin as the first residue of the protein, and then the 6 before it are spacer units, or at the other end of the protein. Hidden units here, just shown 6, 3 output units, because we have helix strand other, 3 states, and this device in fact does not really solve any of these problems. It is a modern device that can learn from more data, and many people had hoped it would do better than simple statistics, but it did not really do better. But it allowed us to systematically addressed some of the issues. And the first one was the issue of beta strands predicted at random probabilities by simply doing a balanced training. Balanced training means at every single time step of training, I present one example of beta strand, alpha helix, and other. The background, you may remember, some of you, that the distribution is such that beta strand is only 20% occurring. Most of the state is the other state, and that is why prediction methods sort of mirrored in unbalanced training. This is of all the correctly predicted residue, what fraction is helix strand other, and that is of all the residues what is observed in helix strand other. And you see these two pies are very similar, and that in fact indicates that the machine learning device simply learned the bias in the data set, which in fact machine learning devices often learn. And this again, now this is an important point, is much, much wider than computational biology. It tells you that machine learning gets out what you have in the data set that you present. But you may not, although that's the best data set you have, you may not really want to have it. In this particular case, we don't want, clearly, we're absolutely clear we don't want a strand prediction at random levels. For that we don't need a master thesis or whatever. In my case, it was a PhD thesis. When I did that, we don't need anybody to spend time on random predictions, right? So often, when you develop something, if you ever encounter the use of machine learning data mining technology, be wary of that. See whether you can find a way to check out whether that problem is one that you face. In this particular case, again, the problem for a developer, if you do not have a lot of background, you may say, well, you know, after all, this actually is declared success. Because all I can do with my machine learning device is to reproduce the data that somebody gives me as truthfully as I can. And by maintaining the distribution, in some sense, that is positively what I do, right? So this is, I presented this here as a problem. I present to you here a simple solution to the problem because I sort of present the computational background or the biological background to the story. In most cases in the future, you will not have that background story. You will not know whether there is anything wrong with it. You have to dig deeper. In this particular case, digging deeper, what is, again, if you look at the pie, what is not immediately visible is the fact that blue is random. So you have to look at different numbers is the message here. Again, here the message is made for computational biology, but more important for you to remember is this is something, the moment machine learning goes off, maybe one day you're going to encounter that and some alarm is going to go on. Again, you can save that, you can change that and predict beta strands at the same level of accuracy. And that is important because, again, in this particular case, it tore down what structural biology had for a decade made as a very convincing argument that beta strand is predicted less accurately because it's less local. And I show it here that the alpha helix formation is determined by hydrogen bonds between residue I and I+4, meaning in between you have 3 residues. So when you look at the window of 7, you have those 3, the hydrogen bond forming in there somewhere. While these here could be between I and I+50. So in a window of 13, I+50 is absolutely not visible. So the hydrogen bonding partner in the window of 13 consecutive residues is not visible. So in that sense, this is less local. And nevertheless, although that is a very understandable and correct argument, this was not the reason, absolutely not the reason why beta strand is predicted less accurately. The local segment of 13 consecutive residues, if trained right, produce the same result. Yes, the biophysical argument is still valid, but it's just wrong. This invalidates the biophysical argument. It may all sound great, but it's not true, okay? And sometimes, again, this is why prediction methods are also referred to as the acidic test of understanding, because this was all very convincing as an argument, and yet before you do a prediction with it, you don't know whether you're right. And ultimately, when I said we don't understand the biophysics of protein structure folding, it's a similar story. We understand a lot about it, but we cannot predict 3D structures, and that means that the essential parts we still don't understand. Okay? So with this, in a machine learning device, and again, it's not that easy to reproduce that in the statistic. People did it with other machine learning devices later. You can then solve already one problem and predict beta strands at a level much higher. The short segments problem in this language of machine learning, this one again, here's a machine, is a neural network. You could have done it with a different device, linear regression, for instance. In the same way, you simply take the first level that goes sequence to structure, secondary structure, one helix strand other, spacer unit, and loses that as an input to a next level. So you have a second level. So that's a sequence to input sequence, output secondary structure, sequence to structure network. And the second one here is a structure, predicted secondary structure to structure. So refiltering. And what it allows you to see is that there are correlations between adjacent residues. That helix, it never will happen that you have something like helix, other, helix, other, helix, other. Helices have to be 4 residues long at least to have an I+4 hydrogen bond formation. And that is exactly what you learn here. And you could not learn that easily in this first level. I showed you a trick how to do that, but it's less trivial. So this way you solve with the machine learning device here the short segment problem, and the way we addressed the problem of limited accuracy was by putting more information into the system, Again, this is the twilight zone story. We have a number of residues aligned, percentage sequence identity. If you have 100 residues aligned and 33 of these 100 are identical, then the two proteins essentially have the same 3-dimensional structure. But if you change 10 in 100, if I change 10 in 100, most likely it would knock out structure. This is because most changes that we can do are most likely not neutral. Not neutral means they will change structure. And typically, change structure typically means they will knock it out in a sense that it is not observable. So you will completely disrupt the thing as an object. I say you put the sequence into a solvent, it adopts a unique 3-dimensional structure. Knocking it out means simply that this statement is no longer true. So by changing residues, you will create a string that will not form a unique 3-dimensional structure. So that will just form a wobbly thing that if you did shine light at different time points, you would observe different things. Okay? You never would see a regular structure or a different regular structure or whatever you would see. For the cell, for life, this implies these things don't survive. This implies why when we collected the data for this curve, actually originally that was Chrysander and Rainer Schneider who did it, but when they originally collected the data, that was any data we could collect is done on existing sequences. Existing sequences are the sequences that survived. And that is a subset of the space of all possible sequences, a tiny subset, in fact, we believe, and a very unique subset. Most sequences would just not exist because they can't. Okay? And that is the issue why if I went to the lab and I changed 10 residues randomly, so people can do a lot with protein engineering. Here in Weinstefan there's a group, Nascara, they do amazing things in terms of protein engineering. They can certainly change more than 10. But if you just randomly picked, if I picked randomly 10 without thinking about it, most likely would do a lot of damage. So the fact that evolution changes, as you say, 67 and still doesn't knock out structure is simply because it evolved to a situation in which it does not have an effect. Things that change structure, effect function, that is not good because that will not deliver the same function for the body, and that body will have typically a disadvantage. So that's why it will not survive. Okay? And that's why, in fact, the neutral observation, or the neutral change, the neutral variant, the neutral change of a sequence, neutral in the sense, does not change structure, that is the least likely, is exactly what we observe. We observe an aggregation in the evolutionary profile. In evolution, what we observe is an aggregation of the least likely event, simply because survival is, the odds for survival are pretty low, and what survives is just a trace of the amazing signals of the unlikely reality in some sense. Anyway, so here this profile tells you something specific about that protein structure. Where you can change what residue is very specific to a particular structure. That means that getting this profile out by taking a sequence, finding a family, looking at that profile, putting that profile into a machine learning device is exactly the way, in fact, this is what I describe here on this picture, into the machine learning device's input. Same as before, this is sequence as input, but rather than having 20, each blob here being 20 zeros and 1, 1, they now become real valued components. It's a simple vector, real valued component that are taken from the frequency of occurrence in that particular family. That method, and here is a sort of more complicated one, but that method clearly does much better. It reaches levels today of almost 80% accuracy. Okay, so now we get into, I began to do that in the last lecture already, the proper comparison of methods. And again, for me, this would be something that I feel it's important for me that you get some feel of what computational biology is, what its challenges are, and this is something that I would hope you would take home out of the lecture partially as a take-home message, because I describe it for computational biology, because I believe it's in this field so important to properly estimate methods, because the way the numbers go. But essentially it's the same for all fields. How do you properly compare methods? And the first question that I asked, I have two methods. One says 60% accuracy, one says 63. And the question really is method B better? In order to address that, the first question you have to ask, before you, typically when people are, well, okay, in TV shows, so in many TV shows today, the best thing to do is just say yes or no, okay? And have a good reason for that as fast as possible. But in science, very often, you have to be a little bit careful about what is it that you are actually asked, and what are the facts you know. And one of the first questions here should be, when I say 60 and 63, it's sort of, you imagine that what I'm talking about is the same score. But that may not be true, right? So the first question would be, are you actually talking about the same score here? And in this particular case, Q3 is the percentage of residues correctly predicted in one of the three secondary structure states. So assume it is the same. Then the next question would be, do they have the same data set, right? Could be that one is using 20 all alpha proteins and one is using 20 all beta proteins. Or that one is using a set of proteins that are very, very similar to each other, and one is using proteins that are not. One is using a set of proteins that are easy, one is using a set of proteins that are difficult. Remember, at some point, some of you may have been in the room when I discussed, typically, whenever you have a number such as secondary structure prediction 65%, typically that's an average of a distribution. And typically, the distribution in this particular case, say it would be 65%, and this would be 65. In some distribution here is the number, the distribution is going up here, and you have some standard variation here, you have some average. And I told you about this intelligent person who said, well, obviously these proteins on that side of the distribution are not performing very, very well, so I'm going to not use them for my test. Because if I threw them away here, if I just say these are bad proteins, there's something wrong with the experimental structure, or something wrong, and my method is only treating the one on the right, I have a much better estimate for my performance. And that again, so ultimately what you need is not only an idea that they have the same data set, ultimately in fact you also want to see the entire distribution. Now things get more complicated. Because looking at, say you compare 10 methods, 10 distributions, you can just visually not easily compare. So then you can look at I don't know how much statistics you had. You can compile the overlap between different distributions, provided they are somehow Gaussian shape. Somehow normal distributions, right? But then things get complicated. So then you look at the area under the curve kind of things. And then you're back to a single number. But in a single number, again, you ignore that there's a distribution underneath. So we have a vicious circle here. And it's not an easy issue. But the first demand here, same score, same data set. Now, same data set is again not solving the issue, right? Because It could have been that both methods used the same data set. Say we have a neural network and a linear regression. And say what we are reporting in the level of accuracy here is only some subset. And they took two different subsets. So the next issue is we have to make sure that in some sense the way they split the data sets is similar. Ultimately what is important here is that each one of them uses every single protein for testing exactly once. Then we essentially know, underneath at least, is the same distribution, without even knowing in detail how the distribution looks. We know that when we see a single number, such as 65, we know in that particular case, if they use the same one, the same number, the same set, and sampled in the same way, that each protein has been sampled exactly once. They can differ in what they do underneath. Okay? So let's just look at this example here. So we stay in this example here. We have one method that does a 99 or 100 times split. So saying that they take 99 proteins for development, one for testing, and then they rotate through 100 times. Another method says what they did is they have a 5-time rotation. So they use in fact 80 for training and 20 for testing. And then they rotate 5 times through. Can I compare these two? And they both claim some number. Say the number that I put up in the method A and B, 60 and 63. Is that okay to do? Okay, I have the fast shot. Yes? Is it? No? Can I see no's? That seems to be a majority. Actually, here we get to the next issue. You guys are right, your intuition is right, and yet in some sense it's wrong. What is it that we want to estimate? We want to estimate for the set that I do not see today. I don't want to apply my method for proteins I know today, right? What I'm measuring here is always true for things I know today. Actually, I don't care about them, but that's all I have today. What I care about are the proteins I have not yet observed, because those are the ones for which my method will be applied. Only for methods for which we do not know the structure, people care about the second-order structure prediction, right? So how do I estimate how the proteins from tomorrow will look like? Well, somehow I have to sample today's set and assume that tomorrow's proteins will somehow look similar to what I see today. Now, if somebody came up with an estimate 63%, and you are the biologist and you test this method on 10 proteins that come out in the future, and the performance will be 70%. That's okay, you'll be happy. If performance, however, will be 50%, you'd be very frustrated with the developer and actually possibly with the field, with anybody who develops second-order structure prediction methods. So what I'm trying to say here is if your estimate is such that it's conservative, so you rather underestimate than overestimate, in terms of wanting to be used and making people happy. In order to make the reviewers of your paper happy, the story is unfortunately the other way around. They may say, oh, come on, we have seen methods like that before, if you can't do better than that. So that is an incentive to publish numbers that look a little bit too high. And that, well, the task is in your hand. Back to the issue. I have one method that samples 5 times, one method that samples 99 times. The method that samples 99 times will tend to have an higher estimate. They sort of squeeze out whatever they can from the training data in the most optimal way. The method that used 5 split did not do that. They threw away information they could have. That means, most likely, they will come to a more conservative estimate. Meaning that if the one with the 5 split is the 63 method, then we can absolutely compare these numbers and say, yes, this is a higher number and it's more conservative. If it's the other way around, it's still the higher number. And we have to ask other questions. But, you know, essentially within the confines of what the developer did, that was quite okay. In that sense, we can compare those two. Okay? Now, the issue of random splits. We cannot randomly split without knowing something about our data set. We need to know about overlap. In the context of secondary structure prediction methods, one thing is clear. When I can predict by comparative modeling, I don't need secondary structure prediction. So for cases for which this is the set of proteins of known structure, I come in with a new sequence, I match in that data set of the PDB. Any case where I match, secondary structure prediction methods will be completely uninteresting. So that tells me about the split here. Between a training and testing protein, if they fit this criterion, so if between training and testing I could apply comparative modeling, then that would be too much of an overlap, because I have another method for that, okay? For the method that you will develop your own in a completely different field, it's not always that trivial. But you have to somehow find a way. Is there anything else that that you're up against, that you have to beat? Is there anything else where you can clearly define what constitutes a too high overlap between two entities in your data set? Here the entities are proteins, and here it's clearly defined. So only when there is no overlap between any of these 100 proteins, then we can really do a random split. Otherwise, we have to make sure that between training and testing, I in fact never can do comparative modeling. Okay. We had that already. And again, so I say that here in detail. In this particular case, too much overlap, anything above, if training and testing are in the blue realm here, then that is too much, and they should not be part. The next issue. One method is 63, one method is 60. Assume we have the same type of split. Assume we have exactly used every single protein once. It's the same data set. It's the same measure. Now, is a difference of 3 percentage points significant? I see no. I see 2 yes, 1 no so far. Can we have more action of heads or fingers? Who says 3 is significant? 1, 2, 3, no. You're just playing with your fingers, right? 2, I'm 2. Who says it's not significant? 2, 3, 4, 5, 6, 7, 8. Who says I don't know? Oh yeah, in this particular case, I usually don't like abstain, but in this particular case, abstain is good. As long as the ones who abstain tell me what question I have to ask.
Size of the data set?
100. You said the size of the data set? 100. I postulate it's 100. That's the example I'm toying with here all the time. So can you ask me any other question? There's another question that is important to ask before we can proceed. Size of the data set is important, but
the whole number of all the proteins that we know or we want to know?
yes, but now you're getting into I will get back to that. All we want to know. I will get back to that. It's not the answer that I'm looking for here, but I'll get back to that. There's something else.
Maybe we are overfitting the 53% data?
We are over-predicting. That in some sense goes back to all we want to know. That is true what you say. And that is a big issue and is a bigger story. Well, it comes under that point.
Be something that has to do with the error somehow.
Yes. What is that something? Yes. Yes, exactly. It's there. You need the distribution. So the number is important and the distribution is important. Typically, say, there's this sort of rule of thumb that's the sigma, the standard deviation here. The standard error is, as a rule of thumb, the sigma divided by the square root of the number, roughly. Where the number is 100 here, in my case, so that's why the number is important. 10, that would be. So if the sigma in this particular case would be 10, so just visually, if that is 65, well, actually visually you cannot. But from the curves I showed you before, so 10 is the ballpark of the standard deviation that we get for secondary structure prediction. Then you would see that plus minus 1 percentage point is what is statistically significant, and plus minus 1 is exactly what we have. The difference of 3 is above plus minus 1. So in this particular case, this would be, at least by 1 standard deviation, a significant deviation. Okay? And by the typical 1 standard deviation kind of thing, a typical 1 standard error, this would in fact be, here's the distribution underneath, considered significant. You say that you can sort of read it off. This is the case for 72, and it's plus minus is roughly 10 percentage points. It's actually exactly that. I thought it was less. Okay, now, I put this down again. Same thing. Now here comes the next question. But method B happens to be 20 years older. So again, we have established same data set, same score, plus minus 3, so it's more than the standard deviation, but it's an old method. Is it still better? And guys, I have to get back to that one. I will go back to that in the end. That's a good point. That's a very, very good point. But that is in fact a complicated point. Now, this is very important. So, if I said in this particular case, 3 percentage points are statistically significant. I did not address the question, is that scientifically making a big difference? Assume it made a big difference, maybe you would argue you can pay more CPU time. But ultimately, whether or not you argue that way depends on the difference of CPU time. If one secondary structure prediction allows you to do that automated in a batch of thousands, and another one allows you to do that 1 a week, then maybe that will become the final argument to say, okay, it's less accurate, the more modern method is faster, or the old one is faster, whatever it is, but I go for the faster method. This is very often an important issue, yes. So if you beat a method by something important in terms of speed, that could make the difference. As long as you can make sure that enough people care for it. So there is somehow, I believe there's always a right middle way between speed and gain. We talked about sequence alignments. Brute forward is dynamic programming. So you align two sequences in a way in which you try all possible combinations. Dynamic programming is in the square of the length of the two proteins, and I told you for aligning 50 million sequences, this is prohibitive. It is too slow for that. So even 20 years ago, when we had much fewer than 50 million, when we just had barely 1 million, even then it was prohibitive. So people turned to fast ways of doing this. And there were two offers. And the first one was called Fast A, and that did a fantastic job at getting the statistics right. It sped it up with good statistics. And there was another one that sped it up in even more with worse statistics, and that was called Blast. In this particular case, Blast took the day. Blast survived. And the reason why Blast survived is what you could do with Blast, you were so fast that you could, in fact, break down the problem. Okay, you couldn't do the dynamic programming on 500,000, but you could do the dynamic programming on 1,000. Okay, Blast did not get the first, not always in those days, we're talking 25 years ago, the first 30, 40 hits were not always right. But in the first 1,000 of Blast, you always had the right ones. So you could afford to have a quick pre-filter of Blast and then do the real dynamic programming thing. And fast A was sort of a path just between these two extremes. And that didn't survive because it did not strike the right balance. So the reason why BLAST survived, although it was less accurate, was because it was so much faster. So this is an important issue. Speed is always something that you have to look in the context of the quality. Now, if we went in a time machine back to the 90s, I'm not entirely sure that it was clear to predict which one would survive. From today, we understand completely why one survived and not the other. But when you are the developer, it's complicated to figure out which one of these two classes you belong to. Anyway, this is just an example. Back to the question. Let's assume that the speed were the same. B is 3 percentage points higher, but it's an older method. Is it still better?
I think B would be better than A because
Yes.
it's 20 years older and it was predicting proteins which are not seen yet.
Oh, that's an interesting point. And that brings us back, yes, I will get back to that. So you make the generalization topic. You say that the new method may have been has a higher probability of doing novel things and sort of learning by heart from the novel things, while the old one really obviously learned something more about a principle, because it could extract for things that happened 20 years later. And that is a credit. And that is good. And I like that view. I try to push you as young people into the view that older means bad, and you didn't go for the trap. So the fact ultimately is this is just about number. As long as there's no difference in time, ultimately it's about numbers and not about age. Okay? What you say here is an interesting argument, but ultimately at the end of the day what counts is the number, and then the answer is yes, it is better. There's no difference in age. And then the question is there any other tests to do? And in some sense, this is what you put your hand on when the pre-release test. Ideally, what you want to do in order to compare two methods is to use proteins that are not similar to anything any of those two has used for their development. And not similar means not only that it's not the same protein, but actually not within comparative modeling distance. So I want to have a set of proteins that are such that when I take the database when those two methods were developed, of proteins of known structure, they could not have done a comparative modeling. They could not have touched my proteins. For those, my new proteins, for them, they clearly would have been new. And now assume that is a set of 100. That's the best thing I can do. This is what I call the pre-release set. Now, the fact is, in the world of computational biology, things move so fast that typically, by the time you are done with your development, there is new experimental information. And that means typically, you can just take the latest release of the database at that moment and look at the differentials, something that came in new to your method, and see how well you do. And compare yourself easily with other methods on that set. And usually that works. And that's a great opportunity. It's a little bit of extra work. Now we get into the next issue. Let's call it how-to cross-validation. And I do see that we don't get into the coffee, that we don't get to the membrane proteins really, except for maybe the movie. So we, in this particular case, we stay in the framework of neural networks simply because I began with them. I have three different methods or three different ways of training neural networks to predict secondary structure. One has 15 hidden units, one has 30 hidden units, one has 45 hidden units. The accuracy is shown in this column on the right. What I show you here are numbers that are derived on the test set. It's not training data, it's only testing data. I put that table into my manuscript, or my thesis, and in the conclusion of the thesis, I say that the best method is using 30 hidden units, because that is the highest number. Let's assume again that 1 percentage point here is the difference that is significant. So let's assume that the values here would be significant differences, okay? But even if they wouldn't, I may say I cannot make that statement reliably, but still this is the highest numerical number, and I possibly can make the statement to 15. Okay, is that proper? Proper conduct? Kosher, as you may call it, or whatever you call it. Is that okay? If you were the referee on that, a reviewer on that, any alarm bell that goes up? So obviously there's an alarm bell that should go up. Tell me why. What's wrong with it?
You're still using the same method, and
Yes.
it has the same quality. We just
Yes.
change the test.
Yes. No. Oh, sorry. No. Yes, indeed, we are using the same method. We're not comparing different methods, but I have one free, in this particular case, I always, when I have methods, develop methods, I have some free parameters. So some of these free parameters are set by the training data, or some of the free parameters I choose in a different context, right? And in this particular case, there's one parameter left, hidden units. I decided that I have to test which number of hidden units with which architecture is best, and in my final thesis, I write that is the table, that therefore the one with 30 hidden units is the best. So right is not intelligently different methods, but is that the right decision for the choice of the number of the open parameters of number of hidden units? Yes, it is true what you said, that those three numbers are derived on the test set, but it's the same data set. Is that right? And I did not use. So training and testing have no overlap. I considered all the issues that I talked about. There's no comparative modeling vicinity. It's a unique data set. In this particular case, you have 300 residues, 300 proteins. So I solved, yeah?
What is this 30 hidden? What is the hidden unit?
Yes, hidden units. That's one way of quickly answering your question is just one free parameter. In the language of the neural networks, so if you had no hidden layer here, all you had is an input to an output, you could essentially only do simple linear decisions, relatively not necessarily linear decisions, but you essentially can introduce only a line in space. Introducing hidden unit blows up the space of your problem into now going into hyperspace. In hyperspace, what you use to differentiate your patterns are hyperplanes. So now you operate in a very complex universe. And that is what you achieve with these hidden units. And in some sense, this is not true. But just imagine this, and again, it's very misleading, but imagine that the more you blow up the number of hidden units, the higher the dimensionality of the problem. That is absolutely true. It's just, it's a misleading image. But in principle, it is true. That's true, right? So the number of hidden units, in this particular case, the type of function you use in the SVM, the radial basis function, or those are free parameters of your machine learning device beyond the connections. Again, what is wrong with this? Something is wrong with it.
Maybe it is overfitting the training set.
Maybe it's?
Because it is more number of free parameters, right? So
that is an interesting point, and that is yeah, that leads over to that one here. Yeah, but it's a different one, slightly different one. Yeah, I will get back to that too. No, something else. Somebody, oh. So the problem is that, and it leads over to the first point here, you're always trying to say something about tomorrow's data set. When you say something about tomorrow's data set, then you have to be very careful that no decision that you make is used based on something that you have today. You pretend that the test set is something you don't know. And now you make a decision based on something that you pretend to not know. You cannot make a decision on something that you don't know. That's wrong with it. Because this means you optimize for today's test set. This means you suddenly take some decision that has to do with something you have in front of yourself. And that is wrong. In this particular case, what you need is a threefold split. Here I call it cross-training. There are, in the machine learning community, there are different words for it. Sometimes people call this training here, this test and this validation. Sometimes they call this test validation, this test, whatever it is. I call it cross-training because then it's immediately clear what I mean. It's part of development. It's part of optimizing some free parameters of your system. And that is part of training, okay? I do it here in this one-third way split. We get back to that in a minute. But in principle, what you have to do is, if for the cross-training 30 is best, then you have to say in your manuscript, okay, I pick 30. If in the end, for the test set, you observe this one, you have to stick to that decision. So that means that now it seems that for your test set, in fact, that would have been the better choice. But you have to propose this one here. Because in your cross-training, that was the right one. And that is then also most likely the better estimate for tomorrow, for this one that we don't know yet. Okay? More fold split. And then actually you really have the guts and say, okay, I made a mistake and I can't explain why the test set in this particular case here is higher, but I continue to assume that this is the best way of doing it, and that's the way I implement on the web. Okay, that brings us to the next issue. Here, how we had it already, how exactly you do this split, in some sense is up to you. It has to do with the amount of time you spent on this problem. So that is back to the issue that we had, 99 versus 1 in 5 split. And that again is the same here. It's up to you somehow how you do it. Let's get back to these two questions here. The first question is, again, what we want to test or what we want to in fact predict are proteins that we don't know yet today. How can we estimate how the proteins of tomorrow will look? What we have today is what we have, so we cannot really use anything else. What we can do is just go a year back, for instance. We can ask if what we had a year ago would have been all we had. And just let's look at what has been added over the last year. Is that very different? If it is, then as a developer at the end of my manuscript, in my conclusion or my discussion, I ought to make that point. I observed that if I used the data set a year ago, my method would have been much worse than what I can do today. Hence, I assume that if I redid it in a year from now, things would be much better. I also assume, given that my estimate here most likely is optimistic. Or most likely will not fully capture tomorrow's proteins. That's an important statement. And then, for the best I can do today, I cannot do better. But this is an important thing for you readers to know. Okay? And for me, as a reviewer on the manuscript, would be an important step to take you more seriously. Just as this one here. If somebody has the guts to do the right thing, that increases the trust. Science is a lot about trust, because the idea is that science is objective, and it is. But very often, at least in this field of computational biology, in math, in certain areas of math, when you review a paper, it takes 3 years. And then you actually go through the math, through every single bit of it. When I review a paper, I do not have the resources to buy myself the, supercomputer that they use for their experiment or for their method and redo everything that took them 3 years to do. It's just impossible. In fact, there are examples, and I will get back to that, where there's one person in the world that has the computer to do this. And that is 10 times bigger than anything else anybody else has. Nobody can verify or falsify that this is right or wrong. Science is a lot about trust, and this helps. Now, this is the best we can do today. We can go back and ask questions that pertain to the consistency of our data set. Do I have indications that if I used only half of my data set, I would get the same result? That would increase the belief that actually at least there is not much change in today's data set. Possibly then, from tomorrow's data set, I would not learn much, and possibly then these estimates are really conservative. That's ultimately what I can do, and if the answer is that in fact I have enough, then the answer is clear. Most likely the answer will be I need more. And then that again is an important statement to make. But again, so I'm talking here about proteins. This is something that you can, for any place where you apply machine learning, try your best to find out. The next issue is, how many parameters are there? Can my model fit? So in the context of the neural network, it's very easy to visualize. The number of parameters, the free parameters are essentially the connections here. So 13 times 21, in this particular case times 6, is this space of free parameters. Okay, then 6 times this one to that one is 6 times 3, but this is a small number. The bigger number is here. Okay, and that defines essentially the number of free parameters, defines the dimensionality of my problem. So, say in this particular problem here, I have, in this particular case, I can't remember, 3,000 input unit, 3,000 free parameters. How many samples do I need? Is 100 proteins enough at all for 3,000 free parameters? So the first statement here is the comparison that I lead your head to take from my mouth is 100 versus 3,000 is misleading. What we are training here is not 100 proteins. What we're training is 100 proteins, each of 200 or 300 residues. So it's 100 times 300. So in this particular case, initially I had 30,000 examples. Because every single residue is one example learned by the network, okay? But now comes back the question. It doesn't sound as dramatic anymore. But I have 3,000 free parameters and 30,000 samples to learn from. Is that a good ratio? And there is no answer to that question. There are, again, machine learning is very much, at this point, cookbook experience. And I'll make another statement about that in a minute. And one cookbook is a factor of 10. I've seen examples where a factor of 10, meaning that 30,000 samples, 3,000 parameters is good. I've seen examples where much less is good. And I've seen examples where much, much more is not good. What does it depend on whether one or the other is true? Well, it depends on the information content in these samples that you present. One aspect is how diverse are the samples? If I gave 30,000 times essentially the same sample, obviously that would not contain a lot of information. How can I measure that? Whether my samples contain enough information. Reduce the sequence similarity, yes. Be careful. So now, let's assume we have our example with 100 proteins. And let's assume that set, there would be no set in there, no pair in there that has more than, say, 20% sequence identity. So they would, by that statement, be divergent, right? But still, it could always largely be the proline, the consistent signal. Okay, there could be a lot of noise in there, and there could be a little signal, and the signal could still be consistently always the proline that breaks the helix. It's not measured in the 20%. Because it's measured on the level of the individual input vector. One sample at a time. And that is not, so yes, at the coarse-grained level, if my 100 proteins would be 100% identical, then all the initial vectors would also be identical. That direction is clear. The other direction is not that clear. If they're not identical, there may still be high overlap in the information. So your point is taken, it helps, but it's not enough. What else can I do?
Maybe we could use cross-validation again and look for the more inaccurate results.
Ah, that's a great idea. But you make my list of things that I have to discuss about. Actually, this one we did, and this one we are doing. Outliers. That's true. We get back to that in a minute. And it is partly an answer. Anything else? So it may come to your head, and in some sense what you said is exactly sort of that thinking behind it, that you can cluster your points. And that if you clustered these initial vectors and you had a high overlap, then there would be little information. If you clustered them and you created many, many clusters, you would have a low overlap. And that logic is right. The trouble is, we cannot measure essentially the overlap directly. If we could think about it, we are thinking about the overlap given the problem. We are asking how informative is a particular vector for the question whether it's a helix strand other in the center. And ultimately, when you think about this question, this is the same as saying I developed a prediction method that answers that question and gives me a prediction. It's exactly what it is. So I exactly created the neural network to answer that question. And now I'm asking what is the overlap, which is a different way of phrasing give me the best prediction. We cannot do that. If we could, we wouldn't need the network. And that is exactly why machine learning, in fact, we go into the cookbooking story. Why these things are black machines that you can somehow open and you can see certain things, as I showed you with the second order structure prediction, that you could tune them to do certain things. But they are black book ultimately because we are at a problem in a situation in which there's nothing else that works. We need a machine learning device to classify. We do not have a better answer, and that exactly is the limit. So now we cannot turn this around and ask, can we pre-filter? And that's why we get into these cookbooking rules where we say 1 in 10. And we as often violate that rule as not. It's as best as we can do. We can, that is getting back to what you said, look at outliers. So we can then, again, look at different ways of looking at this cross-validation distribution, see the distribution of scores, see whether different splits of the data set, in fact, so assume that now I rotated this around 3 times and then the 3 different data sets would have completely different estimates. That would get in the same direction of looking at outliers of my prediction. And that is something, whenever you do any development of code and you produce any picture, look immediately at things that are extreme. Outliers. Typically you can learn a lot about them. Most often, in my experience, you learn about programming mistakes. But very often you learn more than that. And sometimes you can even learn how to improve your method. Okay, with that, we are through with secondary structure prediction, and we go into the field of membrane prediction. And in this field of membrane prediction, we have only just a few minutes left. So when we come to this point, we begin with most likely what is an RNA world. You have floating RNA molecules. They reproduce. They copy themselves, and they begin some form of the early shape of life. And possibly proteins come in to do enzymatic reactions for those, to sort of be slaves for the RNA world. And then we create a soup of something that will become life. In order to create a cell, you need something around it. You need the difference between inside and outside. What do you put around the cell? Well, you need something that is permeable, but in some sense only semi-permeable. So in a medieval village, that is a gate with a gatekeeper. So you have a wall around it that nobody can really get into, and then you have holes in these walls, gates, and they are guarded. And that, in fact, is also what you have in cells. You have holes in the cells, and I'm going to show you in a movie in a moment, and you will see these holes. And many of these holes are gated, guarded. Some of them are not. And when you are not guarding a hole, then essentially what you need is another level, a second wall that is then gated and guarded. Or you need to have some guard directly under the wall, behind the wall. And again, in cells, that is what you have. The way the inside and the outside, since we have water, we have cells swim in water, and in fact inside of cells there's water too. So the thing that you put around are lipids, because lipids are not permeable to water, or only to some extent. And that is exactly the coffee story that I showed you, where you break up the cells, you put out the cell walls, the lipids, and that's why it looks so oily, fatty. Okay, 2 movies, and then that will be it for today, and then we will get next week into...
The cell membrane controls traffic into and out of the cell surroundings. Like all biological membranes, the cell membrane exhibits selective permeability. That is, it allows some substances to cross it more easily than others. This ability of the cell to discriminate in its chemical exchanges with its environment is fundamental to life, and it is the cell membrane that makes this selectivity possible. In this video, you will learn about the structure of membranes and how the outermost membrane of the eukaryotic cell, the cell membrane or plasma membrane, controls the